So this is the IB Bio 2.4 notes. First up, I'm going to be talking about polypeptides, amino acids linked by condensation reaction. Basic unit of a polypeptide protein is an amino acid. Amino acid is carbon-based compounds with a carboxyl group COOH and an amino group NH2. Amino acids bind together to form proteins in condensation reactions and form excess OH and H and form peptide bonds, which are hydrogen bonds. There are 20 amino acids in nature. Anabolism reaction. Synthesize large molecules from smaller and constituent parts. Catabolism reaction. It's a breakdown of large complex molecules into smaller sized molecules. Next up, I'm going to be talking about genes and polypeptides. A gene normally codes for one polypeptide and each polypeptide as a function. Proteins can be made up of one or more polypeptides. The genetic material is transcripted into the ribonucleic acid RNA called mRNA, messenger RNA. This carries the instructions from the amino acid sequence to the ribosome, found in the cytoplasm. Next, I'm going to be talking about polypeptides and conformation. The order of amino acids determine the shape and function of the protein. Conformation is protein's 3D structure. 20 amino acids differ from their R groups, which determine their types of bonds and interactions with other molecules that they can make. This affects how the polypeptide chains fold up in the protein and affect its conformation. Even a single amino acid change can upset the conformation and functioning of a protein. In sickle cell anemia, the glutamic acid is substituted by valine. This causes severe changes to the structure of hemoglobin. Primary structure is the sequence of amino acids in a protein, which defines all aspects of the structure and function of a protein. Secondary structure is the folding of the chains on themselves to form pleated sheets or alpha helices. Tertiary structure is when the polypeptide folds and coils up to form a complex three-dimensional shape. Quaternary structure occurs when in proteins that are made up of two or more polypeptide chains and refers to the way the multiple subunits are held together. Examples are hemoglobin and riboscope. The next slide, I'm going to be talking about proteome. All organisms have unique DNA content, genome, and unique set of proteins, proteomes. Next is examples of protein functions. There are two types of protein functions, which are globular and fibrous. Globular proteins contain or have spherical and tend to affect cell metabolism. It consists of complex polypeptide chains that can be linked to other chains, usually soluble in water because their hydrophobic R groups are folded into the core of the molecule. A fibrous protein is long and thread-like, made up of long polypeptide chains. Insoluble as hydrophobic R groups are exposed, and is found in structural parts of an organism. Functions of proteins contain metabolism, receptors, transport channels, pumps, and enzymes. For the last slide, I'm going to be talking about denaturation of proteins. Denaturation process in which proteins lose their secondary and tertiary structures. The hydrogen bonds formed between R groups of amino acids and amino groups of different amino acids and their shapes are disrupted, leading to enzymes losing their enzymatic properties. Temperature causes denaturation as enzymes lose their conformation, meaning the interaction between certain amino acids will be changed, so that the quaternary and tertiary structures are irreversibly changed. pH causes denaturation by breaking the bonds between the non-adjacent amino acids or between the polypeptide chains of quaternary proteins. Well, that's all there is, and thank you.